today there's KBS session and presentation, therefore I won't be going over it again. And then this is we made a biochemical apps. Most apps, all apps here I'm presenting today was made by Junyoung Lee at PNNL. He's a great colleague and friend. And so our structure of a narrative is also simple. Then we use FTICL data as a molecular formula as input file. And this is generated by the formularity and R-code by Bob Dangzak. And we put this FTICL data file into the so-called staging area in KBase, and then we import that into the created narrative. And then this is the KBase app, also import TSV Excel file. And once it's incorporated into narrative, we can take that as input and we build the biostoichiometric reaction models and from chemical formulas. And then we generate all the chemical thermodynamic properties of chemical compounds, and then also half reactions, catabolic reactions, anaerobic reactions, and metabolic reaction, everything. Also stoichiometric coefficients also show the same different types of reactions. And key output files from there is thermodynamic properties CSV and stoichiometrics oxygen CSV, meaning that this is when oxygen was used and electron acceptor. And then and this file should be downloaded and then uploaded again, and then moved to the staging area and import again. So this is a little inconvenience. However, and to keep them automatically in the narrative, we have to define the objects, but this is something we have to discuss tightly with the KBS team. Anyway, so by uploading this generate data, thermodynamic and stoichiometric reaction, what we can do with it is three things actually. So we run lambda analysis app. This shows that basic purpose of this app is to examine the correlation between lambda and the reaction rates. I have already shown that some of the examples how lambda can be correlated with the reaction rates under what conditions. And also we can use that because the final form of biogeographical model from this theory is that we have a kinetic equations. Therefore, we can perform like a dynamic simulation in a batch reactor or continuous stir tank reactor. So we have a specific apps for that, batch biochemical reaction model and the CSTL reaction model. And this is the output when we run, generate thermodynamic properties and stoichiometric reactions. So you can see here lambda distribution from 0 to 0.3. Mostly it's rare to see that lambda values are greater than 0.3. And then this is their distribution, and then for a specific sample. And then here it's delta G, COX naught. This is typically used for evaluating thermodynamic efficiency, favorability of a compound. Delta G, COX naught is gives free energy for electron donor half reaction as standard condition pH is 0. Also, you can compare the both, but meaning is the same. The lower value means that more favorable, larger values are less favorable. And also automatically generate this Bancroft plot because the data file we have has already classification of each compounds and lignin, protein, and tannin, and so forth. And then this colorful figure is automatically generated in KBase. And also this app generates define lambda beans. Why do I define lambda beans? Because eventually I want to use this output for dynamic simulation. So for example, the Altamaha surface water and pore sample, we have more than 10,000 compounds that have chemical formulas. So simulating 10,000, it is possible, but it takes longer. But sometimes we are not learning a lot by including all. So I think it's a good idea to start from a simplified set or subset of compounds, or we can just define like a representative compounds. So one idea to do that is that we divided lambda based on into a discrete number of lambda beans. In this case, we use the cumulative distribution function, and then depending on the cumulative function, we can divide lambda space into 10 different beans, and each bean contains a similar level of thermodynamic properties with respect to lambda. So lambda 1 contains these compounds, lambda 2, lambda bean 2 here, and bean 3 and bean 10. Bean 10 has a long tail, but actually it doesn't not contains a lot of compounds. The scales are different from here. That actually this is still tiny portion, not tiny, but almost equal number of compounds there, and then they're very comparable. So what we do here is that we identify their average chemical formula from each lambda bean. And then this is plot of their average chemical formula obtained from each lambda bean in van Crevelen plot. So oxygen to carbon and hydrogen to carbon ratio. And these are 10 average molecular formula obtained from 10 lambda beans. Interestingly, these compounds are representative of average compounds. They are not separated by H2 carbon ratio. There are some compounds that are separated here, bean 10, for example. However, most of them are separated by oxygen to carbon ratio. They are more sharply separated by carbon along the oxygen to carbon ratio instead of hydrogen to carbon. And this figure is comparison of the two cases where first we calculate average lambda from each bean. This blue represents average lambda from each bean. In red, this is lambda derived from average chemical formula. So I hope that the audience recognize the difference between these two cases. And you can see here from bean 1 to 10, they are strikingly similar to each other. So we are going to use this lambda value derived from average chemical formula. And my take on this is that this chemical formula defining average chemical formula represent the thermodynamic properties of the compounds in each bean reasonably well. And we have an option for, I'll talk about that again later, but we have an option whether we can use them like original compounds, or we can select them through random sampling, or we can use representative compounds for dynamic simulation. Anyway, so second app is examine the relationship between lambda and reaction rates. And then for correlation, we don't have to specify mu max. Actually, we can divide both terms by mu max because correlation is not affected by scaling factor. And here, and then we now two lumped parameters VH times organic carbon, VH times oxygen, and depending on their values we assign, it shows different correlation with lambda. We can see it's negative correlation when I put here 0.5 for VH organic carbon means that this is moderately limited. Carbon is moderately limited. And then 1.0 oxygen means that this is weakly limited. So it's a carbon limited condition. So simply say carbon limited condition, we can see good correlation between lambda and oxygen consumption rate or biomass growth rate as well. But if you here, now carbon is very weakly limited, here 1.0, and oxygen is moderately limited, then the correlation disappears, and oxygen consumption rate is not really correlated by thermodynamic parameter lambda, meaning that this oxygen consumption rate is not driven by thermodynamics. We can examine their relationship like this, as I discussed earlier. So actually, we can develop based on this relationship to a three-dimensional figure, and x and y axis is VH times organic carbon, oxygen, or their correlation between lambda and respiration rate. But here you can see when the correlation becomes highest, you can see, but June suggested we better look at two-dimensional heat map. This is more effective to quickly understand and when they are highly correlated. So I agree with that. So our app does not provide three-dimensional figure, but two-dimensional heat map. And then here you can see this is weekly limited carbon and then highly limited carbon. And this is weakly limited oxygen and highly limited oxygen. When oxygen is highly limited, we cannot see very good correlation between thermodynamic parameter lambda and oxygen consumption rate. But you can see for oxygen is moderately limited. You can see high correlations observed when organic carbon is severely limited. Actually, you can connect this theoretical analysis with the conceptual model that Vanessa and James talked about this morning. And then you can see increasing organic matter concentration means that this direction actually carbon is less limited. And then in this direction is more limited. When carbon is limited, thermodynamic limitation is a key control parameter that drives biochemical reaction rates. So actually, this is an interesting experience that when I talk to field scientists and our collaborator experimentalists, their observation, sometimes I do not know their recent observation, but still we are talking about the same context. We are really agreeing with each other based on our analysis and experimental observations. And then perform the batch simulations, and we can simulate this ordinary differential equations. And then I have considered here the oxygen dissolution because oxygen is continuously dissolved into media. And then here, this is term that how fast the oxygen can be dissolved into the media. And then this is organic carbon term, oxygen and biomass. But I have introduced here cybernetic control variable U. This is very, very important to make our prediction results consistent and reasonable. If you don't have this, and then you will see that soon, but your prediction on specific growth rate is unreasonably high. Anyway, the cybernetic model has been developed for more than 3 decades by Ramakrishna Group at Purdue University, and then the initial model was focusing on predicting the dioxide growth, trioxide growth, and growth on multiple carbon and nitrogen sources. When then glucose and nitrogen are both present, microorganisms do not consume glucose and xylose together. They preferably consume glucose first, followed by xylose, and resulting growth curve is like that. They grow first, faster on glucose when glucose is depleted, and they switch their enzyme settings from glucose to xylose through lag phase, and then start a balanced growth again. And then we also applied this concept recently to the denitrification process. This happens not only for multiple carbon sources, but also multiple electron acceptors. So oxygen and the nitrate and nitrite, and same phenomenon. Difficult to fit this data and purely based on kinetic models. And this is simulation result, and then by default, we take a 10 random subset of compounds, and this is time, and this is dissolved oxygen, and then while reaction goes on, oxygen content, dissolved oxygen level goes down, and when carbon source is depleted, and it recovers the saturation level soon. And biomass grows, and after that, and slightly decreases by cell death rate. And you can calculate this as a specific oxygen consumption rate and specific growth rate. As I said, without accounting for those cybernetic control, this level becomes higher and higher and becomes unrealistic. And same thing with the CSTR simulation. And then only difference is that we have a feed term, and then inlet and outlet term, and then we have a feed concentration, and then also unreacted dissolved organic carbon and dissolved oxygen is removed from the reactor by this flow. And then D is a dilution rate. This is a operating parameter. And we can also apply the cybernetic variables in this reaction as well. And then results are here. Okay, and then in the narrative, we have downloaded Ondas data from ESS Live, and particularly the Altahama data, and the number of peaks to 22,000, and the number of compounds 10,000. And this is narrative. For those who have KBase account, and then this is already shared in public, and then you may see this, you can copy it and then actually to use it. So this also guides our next tutorial session and in the breakout session. And I set up all distinct questions. Then what each simulation or step-by-step implementation have a specific questions to address. And here, first of all, you have to, as I said, upload your data to your staging area, and then I already imported, but my data is here processed at 48-hour this, and then after putting that in the staging area by drag and drop, and then I can import data into narrative. But if you look at this setting, and then this data is here, job status, it takes 21 minutes, so it takes time. So Atama is particularly big Metabolomics data, and then I already did it myself, and then you can run this model. This is app. This is a beta version. And to find this app, you have to click this R to B, and then you can search this app by typing some of the words contained in this description of the app. And then it really doesn't take long, so 4 minutes. And the results is this. And results are like lambda, as you have already seen lambda distribution. I can click this to see them bigger. Lambda distribution, and then delta G COX naught, and then average value of lambda, average value of delta G COX naught, and standard deviation, median value, and then these 10 different average molecular compounds, and from 10 different lambda beans. And also, you can download all numerical file and CSV, particularly thermodynamic properties file and stoichiometric coefficient file. These are input. You have to download them, and then you can upload them in the staging area again and import here. This is a little inconvenience, but this is a necessary step. And then you have to incorporate, first of all, thermodynamic properties, and then stoichiometric, and then now you're ready to run lambda analysis. And by running this app, you can see, so different, so before that, so I'll show how I set up my input parameter. So actually this is a discrete points of VH times of oxygen, VH times organic carbon. I put 3 distinctive values, 0.1 as strongly limited condition, 0.5 moderately, and 1 weakly limited condition. But you can put more values like a 0.1, 0.22, and 0.9, like a 1. Now, whatever you want to put them here, then you can run. Yeah, I don't remember how long it takes, but may not that long. And then by running this, yes, just 1 minute in this case, because in total 9 cases, and the results is that. So here then how this case is strongly limited, and then both are strongly limited. You may not see correlation. And then best way to do it, just you can look at these heat maps and where they find the good correlation here. And then carbon is significantly limited, oxygen is not very significantly limited. So this case, and you see that a good correlation between thermodynamics and then respiration rate. And then simulate a batch reactor, and this doesn't take long, but number of compounds, this is random sampling. We are not simulating 10,000 compounds initially. So to save our time, then we start from 10, but you can increase this value like 20, but still it's a random sampling. And then also, the beginners do not know necessarily the reasonable values of kinetic parameters, and then this is default parameters, and show then you can see how reaction goes on, and you can change the values around these values, see how the model simulation results will be different. And CSTL is the, but important here, if you click this show advanced, and there are some things, carrying capacities here, but by default infinite because they don't grow a lot, therefore carrying capacity is infinite. And then oxygen saturation water, and it's not exactly 0.25. This is a mole per cubic meter or millimole per liter, but you can change, this is a function of temperature, therefore you can also function of RPM speed, rotation speed, therefore you can put it into appropriate value for that. And volumetric mass transfer rate also is a function of RPM. And then I've taken this value from literature though. And then you can say, oh, simulation ends at 100 hours, and there is number of time steps in between, and then random seed. To get consistent result, we define what random seed we used. And then CSTL is very similar, and then, but there's additional parameters we have to put here. For example, dilution rate. This parameters was not in batch simulation, but now you have to put. So it's important to look at the units. Most of the mistakes that the beginners make would be a wrong understanding of units, and then therefore their values, actual values, are not appropriate because of the units are wrong. So anyway, so CSTL and the results is this. And you can see clearly, it's still not shown clearly, but you can see the one of the carbon is not really consuming initially because they are waiting because of this cybernetic control law. And then this compounds is not preferably used in the beginning, but later is consumed when other compounds values, their concentration become lower. And because of this regulatory effect, their curves are not necessarily smooth, but there are some discontinuous change, still continuous, but very non-smooth change can be observed. And also you can download all output files from here as CSV files or image file. And this is another simulation, but now instead of taking them as like a random sampling, I have used the bean averages to document. So I have taken the averaged molecular compounds from each bean, 10 beans, for example, and then this is 10. And then simulation is you can compare how whether this can be representative of molecules for the entire set of compounds or not. You can then compare the result between two cases. And the same thing here, the CSTL also comparison. Because now I've taken bean averaged the molecular formulas for the simulation as a represented compounds. In our tutorial, the hands-on session, I have generated pre-populated and then made an initial narrative, and they will compare at least 2 time points. And first time point, I have already made a full calculation, and then students are required to see how their simulation results will change as a function of parameter values, and they will perform new calculation using a different time point data. So anyway, also for others who do not attend the breakout session, still please look at this K-based narrative, and then feel free to use them. And if you have any questions, just email me for a discussion and Q&As. I have 10 minutes left, James. Did you get any questions?
Yep, we've got some questions. Are you ready to take some now?
Mm-hmm.
Okay, so can you explain a bit the meaning, physical or biological, of the mu function in the batch model, cybernetic approach?
Yes, that is a regulation. So actually, tomorrow I have a lecture on metabolic model landscape that I have several slides for cybernetic model. This is U. That is my slide. So this is a control variable. So it's meaning that Y then U. So this is a fraction of the gross fractional consumption of among multiple organic carbons. So this is a typical characteristic of microorganisms. If this is a purely chemical reactions, multiple reactions occur simultaneously according to their intrinsic reaction rates. However, biological reactions are not like that. So microorganisms do not consume multiple carbon sources simultaneously, similar to human beings. If you have a multiple different delicious food, you may not eat them all simultaneously. You have to eat what you like the most, and followed by the second you like, something like that. You have to eat them sequentially. Your eating capacity is not infinite. Similarly, that microorganisms cannot uptake them simultaneously. There's a limit. Therefore, this represents well that how microorganisms or microbial community in the environment, they preferably consume that organic carbon and then nutrients that promote their growth the most. The microorganisms are they are not very smart, but their behaviors are very smart. So they know what nutrients will lead to highest growth rate of their biomass. So therefore they choose, in this case, glucose. You can see the slope here is growth rate is higher on glucose. When they grow in xylose, slope becomes slower. Therefore, that's why this organism Klebsiella toca, they take glucose first, preferably, to grow to maximize their growth initially. When glucose is depleted, then switch over to xylose. Same thing here. This U variable will represent their regulatory behavior in taking up the organic carbon and nutrients in an order of promoting their growth.
Okay, let's see. So another question is, what are the batch and CSTR models supposed to represent? Are we modeling microbial populations in rivers or the degradation of hydrocarbons, etc.? Physically, what are we modeling?
So actually, CSTL are often used for mimic really field flow conditions. And then, for example, we can use CSTL often for addressing the problem associated inundation phenomenon, how the flooding will affect there. So it depends on how we set up the equations. And batch reactor, on the other hand, is also often used in the confined domain. And then there's no input outflow much. Still under those limited conditions, those confined conditions, how cell grows and then how the nutrients grow. Also, batch reactor is very useful for characterize the growth rate because there's no input outflow. So we can identify the growth rate from batch reactor and then extrapolate the kinetic parameters for more complex simulations. Therefore, we can synergistically use these settings much simpler than actual systems and in the field, but still we can design them to synthesize our hypothesis or to identify basic parameters.
Okay. Let's see. So we still have a couple more minutes. There's some questions from actually associated with your previous talk. I'll get to them now. So one of them is, how do one deal with inhibitory effects of some compounds on microbial metabolism, such as the presence of oxygen on obligate anaerobes or other toxins?
Right. So actually, we are in the right place because here you can see. We can see here that you can see nitrate is there already initially, but nitrate is not consumed because of inhibitory effect of oxygen. This is exactly how cybernetic control law plays a role. So we don't have to account for the inhibition kinetically because we can do that. I mean, we can add the kinetic inhibition term there, but suppose that your sample contains of thousands of compounds and carbon and nitrogen compounds, we don't know how they inhibit each other. Therefore, cybernetic model is really main goal of using cybernetic control variables to account for them automatically. Why then nitrate is not consumed in this model? Because this model, cybernetic model, identify oxygen is the most preferred electron acceptor. Therefore, it use oxygen first, preferably, and then not using any other alternative electron acceptors until the most preferable electron acceptor is depleted. Therefore, it's automatic. I have not introduced any inhibition term here, but this fitting is very excellent in comparison to kinetic model.
Okay, thanks. We have a follow-up question on the batch reactor versus CSTR reactors. And that is, so would those be modeled experimentally as well and used to define parameters for specific microbes or populations of microbes?
Oh, I don't think I understood the question correctly. Would you say that one more time?
Well, so it's asking, would it be modeled experimentally? So I take that to mean would the reactors be interrogated through physical experiments? And I presume, or rather fit to experimental batch tests. So basically doing actual physical experiments and using the outcomes to define parameters that are tied to specific groups of microbes, maybe in a functional trait way, for example. They didn't say that, but I'm just inferring that. So for example, kind of stuff that Owen Brody was talking about yesterday.
Right. I think it's possible because one of the good advantages of using this batch or CS test is that we assume that we realize we account for homogeneous condition. So we, without having to worry about the transport limitation, because that our observation is the compounding effect of transport limitation and intrinsic biological processes. Therefore, this homogeneous setting really excludes any additional issues caused by transport limitation. And then when we identify like interactions among, still this setting is useful, better than starting from like spatially heterogeneous reactors. That is a more realistic but difficult to identify intrinsic parameters. So probably strategically we can start using that homogeneous reactors batch and CSTL, and then identifying the interaction parameters for binary consortium is much more challenging than single parameters, but we can start from single organism and then increase the complexity by adding additional organism and see how data becomes different, and progressively we can improve the accuracy of parameters. We need designable experimental strategies for that.
Interesting. So Ray is asking, are we going to cover that in any of the tutorials or anything? I don't think we're doing that today. I'm trying to think through the agenda for the rest of the summer school. I don't think there's an element that's tied to sort of linking to physical experiments. Tim, maybe you probably have a better holistic sense of the agenda and all the topics than I do at the moment. Do you have thoughts on that? The extension of more complicated, maybe it's here, sorry. The extension of more complicated transport models is what they're referring to.
Yeah,
I think the answer is yes.
I might touch on that a little bit in my talk this afternoon, but the idea of that, how we would do that. I don't think we're, the pipeline hasn't really developed yet to the point where we're actively doing much more complicated than maybe a 1D column type of simulation. But certainly that's things we're thinking about, and we'll talk a little bit about those.
Okay. Ray says, great, cool. Let's see. So, okay. Oh, we're actually out of time. I will see if we can get to that question maybe later.